Okay. So good afternoon, everyone. It's my pleasure to introduce Dr. Muthye. Dr. Muthye is currently working as a postdoctoral researcher at University of Calgary. He's working on Plasmodium falciparum. Before this, he has over 9 years of postdoctoral experience at State University of Iowa. He did his PhD in bioinformatics from Iowa State University. He has around 5 year teaching experience at the same. Today he'll talk on not just powerhouse of cell. Welcome, Dr. Muthia.
Hi. Yeah, thanks a lot for the introduction. And it's nice to see all of you here. So I'm going to start my presentation. This is the first time I'm using Google Meet. Can uh, can you hear me well?
Yes, sir.
Awesome. All right. So your entire screen. This seems to be the best. Let's do this. All right. Perfect. So slideshow. All right. So excellent. I think the slideshow is visible right now.
Yes, sir.
Awesome. Perfect. Yeah. So today I want to talk about some of my PhD research, talk about my experience doing my PhD in the US, and introduce you to mitochondria, mitochondrial proteomes, and yeah, so and some really cool non-bilaterial animals, right? So, even before beginning, I just want to acknowledge my family and I do this for all my presentations. So, my wife, my mom, dad, and my brother, essentially everything without them is kind of impossible. So, I want to start the presentation by acknowledging all of their efforts in throughout my academic journey. A bit about myself. So I'm Viraj Muthye. Uh, I did my bachelor's in biotechnology from Kelkar College in Mumbai. That's where I am originally from. Then I did 1 year of my master's in biotechnology from Muthye College in Mumbai. So I didn't finish my MSc there because in the middle of it, I got accepted for my master's in biotechnology at Northwestern University. And this in my master's, it was basically on industrial biotechnology. So I was looking at biofilm characteristics of a really interesting microorganism, Legionella pneumophila. It causes Legionnaire's disease. And I essentially was trying to grow biofilms of Legionella pneumophila with all different microorganisms within bent scale bioreactors, trying to optimize conditions and looking at how they persist with other organisms in its biofilm. So it was all experimental work in my master's. And this was the paper which came out from it. After that, I got a PhD in bioinformatics and computational biology at Iowa State University. And following that, I did 2 years, but little less than 2 years of my first postdoctoral researcher position at the same university in the US. And I'll talk about my PhD research in much more detail, but I just want to talk about what all I did throughout my academic career. So in my first postdoc, I looked at the evolution of a particular gene family, this Muthye family of DNA repair proteins in animals. And then currently, I am a postdoctoral researcher in the University of Calgary in Canada. And I just started this year in October. And in here, essentially, I'm currently working on a project which is looking at molecular mimicry in Plasmodium falciparum. But my research goal for my postdoctoral research or position is to sequence the genome of a really interesting animal, which is Myxobolus cerebralis. It's a myxozoan. And I just want to introduce this animal to you initially because all of my research throughout my PhD in my postdoc focuses on this really interesting group of animals called non-bilaterian animals. And Myxozoa, this is a non-bilaterian animal, Myxobolus cerebralis. And what happened in 2016 is nearly 180 miles of the Yellowstone River, they had to shut it down. All activities had to be uh, shut down, like fisheries, everything, all recreational activities. And that's because tens and thousands of fishes were dying. And turns out that this tiny microscopic animal over here, these myxozoans, these were responsible for this very high mortality rate. Now, myxozoans are animals, they are in phylum Cnidaria. This phylum has uh, the other relatives of this in the same phylum include jellyfish, corals, sea anemones, so animals which we are slightly more familiar with. But these are small, tiny microscopic animals, obligate endoparasites of fish. And by closing this river for multiple weeks, the total economic loss was 500,000 US dollars. And so it is economically, it creates a lot of problems. And my research project, I'm trying to sequence the genome of this myxozoan over here. And the reason I spent some time introducing this animal is because it belongs to a group of animals my entire PhD focused on. In today's kind of presentation or talk, I want to talk very briefly about what I did in my PhD, a bit about what I learned about my PhD experience in the US, and just basically letting you know that all of this happened uh, with me not knowing any programming before I started my PhD, kind of driving the point that a very important point about doing PhD in the US is that a lot of skills which you think you may not have now, if you do a PhD in the US, I believe that there is a lot of encouragement and chance for you to learn those skills and finish your PhD. All right, so my PhD was basically in the US. My PhD was looking at the evolution of the animal mitochondrial proteome. And that's kind of the talk for talk. That's kind of that's why I called it not just powerhouses of the cell, because mitochondria are really interesting and so much more than just uh, organelles that create energy to keep us alive. So, what are mitochondria? Mitochondria are double membraned organelles. They are found in most eukaryotic cells. Their origin can be traced to an endosymbiotic event between a bacterium and an archaean. And while they're most well known for their involvement in ATP production by oxidative phosphorylation, uh, the list of their functions extends well beyond energy metabolism. This includes stuff like iron sulfur cluster biosynthesis, amino acid metabolism, lipid metabolism, signaling, apoptosis, and so on. Uh, Really interestingly, mitochondria possess their own genome, the mitochondrial genome. The human mitochondrial genome was the first animal mitochondrial genome to be sequenced. It's a small, compact, circular molecule with just 37 genes, a single large non-coding region, and a modified genetic code. Currently, over 6,000 animal mitochondrial genomes have been sequenced, and over 97% of them are from a group of animals called as bilaterian animals. So these are animals which include us. And majority of bilaterian animal mitochondrial genomes really do resemble the human mitochondrial genome. So they're not very diverse when it comes to gene content, genome architecture, and so on. So they're kind of similar. But bilateral animals represent just one of the five major branches of animal phylogenetic tree. The other four, that is phylum Porifera, that is sponges, phylum Cnidaria, so this includes the jellyfishes, coral, sea anemones, the tiny myxozoan I introduced you before, phylum Ctenophora, and phylum Placozoa. These form a group of animals. They are called as non-bilaterian animals, roughly meaning that they lack a bilateral symmetry, but it's slightly more complicated than that. But these are very interesting animals, and I'll talk about why I was interested in looking at their mitochondria instead of the bilaterian mitochondrial genomes. Something really interesting about mitochondria is that the analysis of mitochondrial genomes is just one aspect of understanding mitochondrial evolution. The other very important aspect of understanding how mitochondria evolve is to study the mitochondrial proteome. So, what is the mitochondrial proteome? The mitochondrial proteome is a set of proteins which functions in the mitochondria. So I just introduced you to the mitochondrial genome, which encodes roughly just around 13 proteins. But in mammals, over 1,500 proteins function inside the mitochondria, and so of which only 13 are contributed by the mitochondrial genome. The remaining 99% of proteins which are which actually carry out all these important functions in the mitochondria, they are actually encoded by the nuclear genome. They are synthesized on cytosolic ribosomes, and then they have to be correctly imported into the mitochondria for correct mitochondrial function. And so in my PhD, I looked at the evolution of this mitochondrial proteome across animals and developed two tools to facilitate further analysis of the mitochondrial proteome. And like I said, over 99% of proteins which are functioning inside the mitochondria have to be successfully imported into the mitochondria. There are multiple pathways which we know which exist for importing these nuclear encoded mitochondrial proteins. But by and far, the most of these proteins are imported by just one pathway. This is this mitochondrial targeting signal pathway. And in this pathway, proteins have a short targeting signal at their end terminus, which directs them to the mitochondria. So the protein goes through the outer membrane of the mitochondria, then through the inner membrane, then inside the mitochondrial matrix. Once inside, peptidase cleaves off this targeting signal, the protein folds and starts functioning. In yeast, around 70% of all mitochondrial proteins are imported by this pathway. So it's kind of really important to look at this pathway while studying how the mitochondrial proteome has evolved. Uh, Also, something interesting about these targeting signals, which are found in this pathway, is that they have certain shared characteristics, and there are many tools which can look at the sequence of a protein, especially towards its end terminus, and can predict whether it has a mitochondrial targeting signal or not. So this targeting signals are typically found in the first 90 to 100 amino acids at the end terminus of these proteins. They're enriched in amino acids like arginine, serine, and alanine, and they're depleted in negatively charged residues. But most importantly, they form this kind of alpha helix structure with hydrophobic residues on one face and positively charged residues on the opposite face, an amphipathic alpha helix structure. And so these properties are actually, there are multiple tools like Target P, mitophages, and I'll talk about them later, but they can actually predict whether a protein has these mitochondrial targeting signals or not. All right. So while I said that there are 6,000 mitochondrial genomes which have been sequenced, the number of mitochondrial proteomes that have been characterized for animals is very, very low. It has not kept up pace with the number of mitochondrial genomes which have been sequenced. Sorry. So mitochondrial proteomes have been characterized for animals, protists, plants, and fungi, so all major eukaryotic groups. But in animals, only 4 animals have complete or, well, near complete or highly incomplete mitochondrial proteomes, which have been experimentally characterized. So you have human, mouse, which have been by and far the most well studied. Then you also have C. elegans and Drosophila, so all model organisms, bilaterian animals over here. And mitochondrial proteomes, they basically have been characterized by multiple approaches, but which can be broadly grouped into 3 categories. First is purely experimental characteristics, experimental techniques like mass spectrometry or microscopy. Second are purely computational techniques like predicting those targeting signals I just mentioned, looking at orthology search, so you're looking at orthologous proteins to known experimentally known mitochondrial proteins, phylogenetic profiling, co-expression analysis, and so on. But by and large, the most successful approach has been an integrative approach. So essentially, an approach where you combine both experimental and computational techniques to predict these mitochondrial proteomes. And the most really interesting data sets from these integrative characterization techniques are something are called as Mitocarta and IMPI. And they use a bunch of different experimental and computational tools to generate a really high confidence level of mitochondrial proteins in human and mouse. And for IMPI, it's human, mouse, and rat. So these are machine learning based approaches which uh, combine different features from both experimental and computational analysis. And we use these mitochondria proteins a lot in our analysis. All right. So, like I mentioned, over 6,000 animal mitochondrial genomes have been sequenced, and over 97% of them are from bilaterian animals. And majority of these bilaterian genomes, they resemble the mammalian mitochondrial genome. So our human mitochondrial genome, this tiny, circular, compact molecule with a very low number of genes and a single large non-coding region. But the story is really different for non-bilaterian animal mitochondrial genomes. So again, all extant animals can be grouped into five major groups of animals. That is, multiple phyla from bilaterian animals. This includes humans here. Then you have four phyla of non-bilaterian animals. We have Cnidaria, Ctenophora, Porifera, and Placozoa. And my lab kind of focuses on these animals. Well, focuses on these animals at Iowa State University. So this image basically shows the number of described species versus the number of complete mitochondrial genomes which have been sequenced, which of course is very high for bilaterian animals. But what I want you all to focus is, sorry, so have to ask this because I'm using this meet for the first time. Can everyone see my mouse pointer?
Yes.
Awesome, perfect. I thought I was just pointing for myself, so perfect. So essentially, what you have is a rough phylogenetic tree of animals, and each box here represents a departure from the typical animal mitochondrial genome, which is what we have, like a circular, compact molecule, basically. And each color represents a different change. So let's take a look at just one group of sponges. So phylum Porifera is basically sponges, really cool, interesting marine and freshwater animals. And a really interesting group of them are calcarean sponges. And so they have these three boxes here. A black box means that all of their mitochondrial genomes are not circular, but they are linear. So all of them are made up of linear chromosomes. This maroon box means that instead of just one circular molecule, it's a multipartite genome. So all of the sequenced mitochondrial genomes from this group of sponges are made up of many linear chromosomes. Additionally, there are multiple genetic code changes in each of these calcarean sponges as well. So, to give you an idea, one of the genomes which we are trying to sequence now is made up of nearly 150 linear chromosomes, of which most of them have no coding material on them. Now, compare this to the human mitochondrial genome, which is just one circular molecule with with basically really compact. So you have gene after gene after gene after gene and so on, and a large non-coding region. So these mitochondrial genomes are very diverse in non-bilaterian animals. And so previous studies have shown that non-bilaterian phyla encompass most of the mitochondrial genomic diversity observed in animals. The mitochondrial proteome had to accommodate these changes because the proteins which are encoded by the mitochondrial genome, all of them function in energy metabolism, in oxidative phosphorylation, those 13 proteins. They have to interact with proteins which are successfully imported into the mitochondria. So they are interacting with each other. And so all of these changes to the mitochondrial genome we hypothesized that the mitochondrial proteome had to accommodate these changes. It had to evolve. And so, to get a broad image of animal mitochondrial proteome evolution, we need to characterize mitochondrial proteomes from these non-bilaterian animals as well. And this was the motivation for kind of my overall question for my PhD, is that is the diversity in the mitochondrial genomes of non-bilateral animals reflected in their mitochondrial proteomes? And that's just a very rough but overall question for my PhD. And so, in my PhD, essentially what I did is initially I carried out the first in silico comparison of mitochondrial proteomes, predicted mitochondrial proteomes in non bilateral animals to show that they are very diverse. Next, I performed the first comparison of existing experimentally characterized mitochondrial proteomes from bilaterian animals, so human, mouse, C. elegans, and Drosophila. And we were looking at what could be the mechanisms behind this observed big variation in the size of these mitochondrial proteomes. And finally, I developed two bioinformatic tools to facilitate further research of mitochondrial proteomes in animals. First was a database which houses existing comparative knowledge on animal mitochondrial proteomes. And the next is a machine learning tool which predicts mitochondrial proteins from in animals, basically. So that's kind of the whole overarching goal and approach in my PhD. So this was like the first, and I'll basically be scanning over some of the interesting results. So, and if you have any questions, you can just stop and ask me, or I'll basically have my email address at the end of the presentation in case you want to talk to me later about it. But the first part of my PhD was to perform an in silico comparison by predicting mitochondrial proteomes in non-bilateral animals. So, like I mentioned before, none of these non-bilaterial animals have experimentally characterized mitochondrial proteomes. So, to kind of fill this gap, knowledge gap, a practical approach is to use computational tools to predict mitochondrial proteins within these animals, and then look at how it's evolving across overall metazoa. And for that, essentially, we use transcriptomes as our input data sources. The transcriptomes which were used in this study, we made sure that they represent the major classes from all the four non-bilaterian phyla. And then from these candidate transcriptomes, we essentially predicted proteins using transdecoder and used two approaches to identify mitochondrial proteins. The first approach was an orthology search. So we have mitochondrial proteomes from human, mouse, and yeast over here, which have been experimentally characterized. So we took those as reference mitochondrial proteomes. These are mitochondrial proteomes which we are very confident that these are mitochondrial proteins. And then in the first approach, we identified orthologs of each of these mitochondrial proteins in the transcriptomes of these non-bilaterial animals. In the second approach, we didn't look at orthology, but we looked at proteins which may have those targeting signals. So just a quick recap of those targeting signals. A majority of proteins to the mitochondria which have to be imported, they have a short targeting signal at their end terminus, which has certain shared properties, and there are tools which can predict their presence or absence in proteins. So what we did is we used two very widely used tools, TargetP and Mitophates, and identified proteins which have those targeting signals at their end terminus. So now we have proteins which are orthologous to known mitochondrial proteins in human and yeast, and proteins which have mitochondrial targeting signals. And we did this for each transcriptome from these non-bilaterial animals. We combined those results, removed possible contamination, and then whatever remained, that was our inferred animal mitochondrial proteome. And then we compared them. And so essentially the entire analysis can be summed down into this one image from the paper. So what you see here is that on the y-axis, you have species. So the black bars are the reference mitochondrial proteomes for human and yeast, Saccharomyces cerevisiae over here. And these colored bars in the middle, these are the inferred mitochondrial proteomes. So for each of these transcriptomes, they were put through this entire pipeline, and these were the predicted mitochondrial proteomes identified. So what do these colors mean? This green bar here, this refers to proteins which are orthologous to known mitochondrial proteins, but they do not have a targeting signal. This purple bar at the top, these are proteins which have those N-terminus mitochondrial targeting signals. They are not orthologous to any known mitochondrial protein in human or yeast. And these tiny portion slice in the middle, this orange slice, these are identified by both approaches. That is, they are orthologous to known mitochondrial proteins, and they also have a targeting signal, which is actually very surprising because this number was very low in these mitochondrial proteomes. But the kind of overarching message from the first paper was that these non-bilateral mitochondrial proteomes vary greatly with respect to both size and content. So there's a large variation in size, not just between these phyla. So you have phylum Porifera, Cnidaria, Ctenophora, Placozoa, and Quinoflagellates. These are outgroups to animals. So this variation is not just seen between these phyla, but also within each phyla. So for instance, in sponges, this number ranges from just over 500 in this glass sponge, Hylonema populiferum, to over 2,100 in this calcarean sponge, Leucosolenia complicata. We found that most of these mitochondrial proteins were identified by orthology alone, and that very few number of these proteins actually had targeting signals in them. And further research, we essentially were wondering whether because we were kind of surprised why are so few proteins having this targeting signals across these animals. And so we took proteins from TCA cycles, that's a Krebs cycle enzymes. These are very well conserved enzymes across all animals, and all of them have targeting signals in animals. Well, in humans, basically, human mouse, they have targeting signals. And when we aligned those proteins, we actually saw that this loss, this kind of absence of targeting signal is not because of a technical reason. That is, it's not because mitophages or target P, these predictors were not able to identify it, but it was actually because when we aligned those proteins, we couldn't see a targeting signal at their end terminus. So I want to give two examples from these really interesting outliers from this analysis. First is the largest inferred mitochondrial proteome. This was from this calcarean sponge, Leucosolenia complicata, and it has a very strange mitochondrial genome. This is the sponge over here. It also looks pretty cool, but strange. So all genes in its mitochondrial genome are located on individual linear chromosomes, and there are at least 50 extra mitochondrial chromosomes with literally nothing on, no coding sequences in them. It had the largest predicted animal mitochondrial proteome, 2119 proteins. And we looked further at these proteins. So we were looking at what is causing this increase in the number of proteins. We found that it had a very high number of duplication in mitochondrial proteins, which were present as single copy in human. And that there are a very large number of proteins which are not orthologous to known human or yeast mitochondrial proteins, but they do have a targeting signal. So they could be sponge-specific mitochondrial proteins. But more interestingly, we also found nearly 87 cases of neolocalization following gene duplication. Now, by this, what I mean is you have one gene, which is this ARK72 gene in human, represented by one protein. It underwent duplication in Leucosolenia complicata. So there are now 1, 2, 3, 4, 5 copies, and only one of them acquired a targeting signal. So it is one of these copies acquired a targeting signal in human. So this is a non-mitochondrial protein in human, which underwent duplication in this sponge, and one of the copies acquired a targeting signal. On the other hand, the smallest animal mitochondrial proteome of just 454 mitochondrial proteins belonged to a myxozoan Kudoa type from phylum Cnidaria. It's an obligate endoparasite, and it is known to have lost several well-conserved genes, and it has a very low number of proteins which have a targeting signal but not orthologous to known mitochondrial protein, just 91 compared to 686 in this sponge. And it has a very unusual mitochondrial genome as well. Only 8 protein coding genes, a very high rate of sequence evolution, and 3 open reading frames of no unknown function. Plus, it is these genes are distributed across 2 circular molecules instead of just one, as is the case in the human mitochondrial genome. So, next, what we were interested in is, well, that was in the first, we predicted mitochondrial proteins in non bilateral animals, and we compared them, and we found that they are very diverse. A lot of these proteins don't have targeting signals, but we couldn't answer a lot of questions with that data set because those were all computational results. But we realized that we do have 4 data sets of experimentally characterized mitochondrial proteomes from bilaterian animals. So from human, mouse, C. elegans, and Drosophila. And we could actually use those mitochondrial proteomes to ask more interesting questions about mitochondrial proteome evolution. So one of the things, really interesting things we saw is that while they have near identical mitochondrial gene content, so while their mitochondrial genomes are near identical, their mitochondrial proteomes, again, are quite diverse with respect to size and content. For instance, the Drosophila mitochondrial proteome, these are all experimentally characterized mitochondrial proteomes, has only 838 proteins compared to 1700 in humans. And so we we essentially asked these questions. These were the questions which we asked, which is what is the mechanism behind this observed variation in the mitochondrial proteome size? What is the functional significance of this variation? And how much of this variation can be attributed to the gain and loss of mitochondrial targeting signals? So, how much of this can be attributed to the gain and loss of mitochondrial targeting signals? And what are the functional implications of this big size difference between the mitochondrial proteomes? And to this end, we hypothesized certain mechanisms which could be possible. So, first was that this variation in the size can be explained by either duplication and or loss of ancestral mitochondrial proteins. It could be explained by neolocalization of ancestrally non-mitochondrial proteins to mitochondria. So by that, what I mean is non-mitochondrial proteins gain a targeting signal and are now targeted to the mitochondria in a particular animal. Or the third is simply evolution of novel mitochondrial proteins. And because these are experimentally characterized mitochondrial proteomes, we could actually ask these questions. And to that end, what we did essentially is we used the 4 experimentally characterized mitochondrial proteomes, which are human, mouse, C. elegans, and Drosophila. These encompass the 2 main bilateral groups. So these are both from protostomes and from deuterostomes. We also used experimentally characterized mitochondrial proteomes from 2 outgroups. First is the protist, Acanthamoeba castellani, and second is yeast. So they again both have experimentally characterized mitochondrial proteomes and are good outgroups. So what we did is we took their predicted proteins from their genomes. We identified groups of orthologous proteins using protein ortho. So this was kind of the orthology tool which was the best at the time when I did this. There are many more better have come since then. But that time I used protein ortho to identify groups of orthologous proteins from all these different animals and the outgroups. We identified targeting signals in all these proteins using mitophates and target And then for any sort of functional analysis, we used various different tools like PantherDB, WormMine, FlyMine, StringDB, and so on. But I do want to take some time to talk about how we try to answer these questions. And to answer these questions, we use this decision tree. And this decision tree was used to divide the mitochondrial proteomes into four categories for all the four animals. All right. So, like I mentioned before, we identified groups of orthologous proteins between human, mouse, C. elegans, Drosophila, and the two outgroups, Acanthamoeba and yeast. We looked at each outgroup, which contains at least one animal mitochondrial protein. And then we looked at what else is inside that ortho group to divide the animal mitochondrial proteins into four categories, right? So the first is that we looked at each animal mitochondrial protein and asked this question: is the protein present in an ortho group? If the answer is no, which means this animal mitochondrial protein does not have an ortholog to any other species in our analysis. So it was a category 4 or a species specific mitochondrial protein. But if it was present in an orthologous group, that is, it had an ortholog to at least one other protein in our analysis, we looked closer at that ortho group. And we asked this question: does this ortho group have an outgroup protein? That is there a protein from Acanthamoeba or yeast inside here, a non-animal protein? If the answer is no, it means that this animal mitochondrial protein is not orthologous to any outgroup protein. So it's a novel animal mitochondrial protein or a category 3 protein. But if that group of orthologous proteins had an outgroup protein, then we looked more closely at that outgroup protein. And we wondered, is that outgroup protein mitochondrial? If the answer is no, then this animal mitochondrial protein is a really cool example of neolocalization or a category 2 protein. So, an ancestrally non mitochondrial protein in the outgroup now is targeted to the mitochondria in this animal, which means it underwent neolocalization. So, it's a category 2 protein. And finally, if both the outgroup protein and the animal protein both were mitochondrial, then it's a very well conserved ancestral mitochondrial protein or a category 1 protein. So we took each known mitochondrial protein from these animals, four animals, and put it through this decision tree and divided all these proteins into four categories. And then we looked at the distribution of these four categories from these four animals. And so, what we saw is that when we analyzed all the four categories of mitochondrial proteins in more detail, essentially, to study the conservation and evolution of the animal mitochondrial proteome, what we saw that this analysis paints a picture of a very dynamic organelle with a very small number of conserved ancestral mitochondrial proteins. So just 205 mitochondrial proteins are ancestral conserved, and a large number of lineage specific additions and deletions. So you see a large number of lineage specific additions and deletions over here. So you see these are novel animal proteins, 494 mitochondrial proteins from human and mouse don't have orthologs to C. elegans, Drosophila, or the two outgroups. Meanwhile, 275 and 124 uh, mitochondrial proteins from C. elegans and Drosophila, respectively, don't have orthologs to any other protein in this analysis. So I realized I forgot to explain what this figure is. In this figure, sorry, you see a Venn diagram of mitochondrial orthologs from human, mouse, C. elegans, and Drosophila from each of those four categories which I mentioned before. And so this is the ancestral category of mitochondrial proteins. Very low number of ancestrally conserved mitochondrial proteins. And these are novel proteins, novel animal proteins, proteins which have no ortholog to the outgroup proteins. And so this is also a very high number. And finally, category 4, these are species specific mitochondrial proteins. So these are proteins which don't have an ortholog to any other species, not even in the remaining three animals. More interestingly, we also identified a large number of neolocalizations in mammals. So this 164 number here essentially means that these are mitochondrial proteins in human and mouse, but but these are orthologs of non-mitochondrial proteins from either C. elegans and Drosophila or both. So this was also fairly interesting. And while we did look at the functional analysis of these differences and found interesting results, like for instance, we found that a lot of these 494 novel animal mitochondrial proteins in uh, mammals were involved in signaling pathways to kind of consistent with this higher organization of mammals compared to C. elegans and Drosophila. Right now, I want to talk about possibly the most interesting point from the paper, which is how much of this variation could we attribute to the gain and loss of targeting signals. And this was interesting because we kind of saw that the gain and loss of targeting signals did not play a significant role in mitochondrial proteome evolution. And this was interesting because majority of mitochondrial proteins are actually imported via this pathway. And even before I go ahead, it's important to know that none of these mitochondrial proteomes are complete. Our understanding of targeting signals, even this far, is not complete, which kind of makes the field really interesting. But we did see what we see here in this figure are these category 1 ancestral mitochondrial proteins, category 2 the proteins which are underwent neolocalization, then category 3 proteins, novel animal mitochondrial proteins, and category 4 are species specific mitochondrial proteins. So essentially the same four species, the categories of proteins, which we were talking about before. And on the x-axis, for C. elegans, Drosophila, human and mouse. And on the y-axis, you have the percentage of proteins with targeting signals. And so what we see here is that nearly 70% of these ancestrally well-conserved mitochondrial proteins have targeting signals. Meanwhile, none of the other category proteins have a high number of proteins with targeting signals. Now, category 2 is the neolocalized proteins. These are proteins which are non-mitochondrial in outgroup, non-animal outgroups, but mitochondrial in these animals over here. And we see that this did not happen primarily by gaining a targeting signal. And a large number of novel and species-specific mitochondrial proteomes also lack these targeting signals. And so this suggests that there are alternate targeting pathways or non-canonical targeting signals which actually facilitate the import of a sizable portion of the animal mitochondrial proteome. And more importantly, if we use a targeting signal prediction only approach to identify mitochondrial proteins, which is kind of done quite frequently, we will get back proteins which are very well conserved, but actually miss out on proteins which might be novel in that animal. And so we should kind of use integrative tools to predict these mitochondrial proteomes. And that was kind of an important take home message of that analysis. And so finally, now that I did an in silico analysis of non-bilateral animal mitochondrial proteomes, so these were animals when there was no experimental data available. And then we looked at existing experimentally characterized mitochondrial proteomes in bilateral model animals. Uh, the next step was that we developed two tools to facilitate further research of these mitochondrial proteomes. And there actually is a very important need for these two tools. So first is we have a database called as the Metazone Mitochondrial Proteome Database. And in this database, we reanalyzed the existing mitochondrial proteomes from human mouse, C. elegans Drosophila, even the two outgroups, Acanthamoeba castellani and Saccharomyces cerevisiae. And using an R-Shiny framework, we created a database to house those results. So we have orthology results, targeting signal prediction results, protein domain content, and also gene ontology term analysis of these mitochondrial proteins in one database. And why is it important to house it in one database? Currently, data on the mitochondrial proteomes of these four animals is badly scattered across multiple databases. And if you want to do a comparative analysis of existing mitochondrial proteins, compiling the data and organizing it is fairly tricky. And so, for instance, just to give you one example, the C. elegans mitochondrial proteome is present as a flat file, a Word flat file. That's it in the results section. And so that's so essentially, if we have it in one place, it will, uh, one second, it will help facilitate comparative analysis of animal mitochondrial proteomes. And let me see, I am trying to load the database, but uh, this seems to take some time because it's housed in the US at Iowa in my previous server at Iowa State University. Um, minute. So let me see if I can share this tab. And so this is the database, basically. I hope that the sharing is changed. And so we organized this database into 4 tab sets: orthology, targeting signal, domain, and gene ontology. And this is again for facilitating comparative analysis of experimentally characterized mitochondrial proteome. And there are many each of these tab sets can be used in multiple different ways. So one way I can show for one, I want to show one example of each, a little bit of each of these. So let's say you want to download one protein, let's say fumarate hydratase. You have fumarate hydratase protein which you want to download from all these different animals and then analyze it. All you need to do is start writing the name of the protein, and it will show up over here. And then all you need to do is enter the ortho group number from the analysis, and it will show you how many proteins were identified. You see there is a duplication in Drosophila melanogaster over here. There is, it tells you the predicted mitochondrial localization, protein name, target P prediction, mitophages prediction, so whether or not it has a targeting signal. Uh, is it predicted to be mitochondrial by Go analysis in the Human Protein Atlas? Is there any GFP study which confirms that it is mitochondrial or not? Is there any mass spec analysis which has predicted that whether it's mitochondrial or not? Yeah, and which tissue is it found in? What's domain composition? So it has a bunch of this different information, which you can either download sequences or play around with this data. You can download my proteins which have a particular domain. So let's say you want to download a domain, all mitochondrial proteins containing this mitochondrial carrier domain. All you need to do is start entering the mitochondrial protein domain, name that PFAM name. It will not only show you how many proteins have this domain across the species, but also how many combinations is it present in. And here you can toggle with some of these fields. So if you want to download only mitochondrial proteins, you can just click only mitochondrial. It has adjusted itself, and you can download the sequences table or anything. The last analysis I'll show here is, let's say you want to identify mitochondrial proteomes which are, as of now, not identified as mitochondrial. So let's take a look at only non-mitochondrial proteins in human. So right now, of the human proteome 19353, non-mitochondrial proteins are recognized, and you can actually select proteins which have a targeting signal predicted by mitophages and a targeting signal prediction by target P. So you actually have 285 non-mitochondrial proteins which have a targeting signal predicted by both mitophages and target P. Again, kind of really interesting candidates for further experimental research. You can further look at whether there is any mass spec data which says that it's mitochondrial. And now, in fact, there are 21 human proteins which are right now thought of as non-mitochondrial, which have a targeting signal. And there is at least one mass spec study which has identified them as mitochondrial. So these are some of the ways this database can be used. I'm going to go back to the previous presentation. And the last tool, last thing which I want to talk about again was that last thing was that we developed a machine learning predictor which helps predict mitochondrial proteins in animals. So by that, we call it mitopredictor. And what mitopredictor does is that it takes a set of proteins from a transcriptome or a genome, and then it uses three different sources of information. First is orthology search. So orthologs to known mitochondrial proteins are identified. Next is mitochondrial targeting signal prediction. It identifies targeting signals in these proteins. And step four is what we came up with, is we look at the protein domain content using PFAM in it. And we use extract features from each of these three steps. We then trained a random forest classifier, and we showed that mitopredictor could predict mitochondrial proteins better than the prevailing best mitochondrial protein predictor at the time, which was subcons. I won't go into this into too much detail. You can always talk to me uh, about this, ask me questions. But the whole point of MitoPredictor was not just to improve prediction capability, it was also to facilitate comparative analysis of mitochondrial proteins. So it was to make this analysis easy and interpretation of this easier by biologists as opposed to bioinformaticians. So what MitoPredictor does is, after it identifies the predicted mitochondrial proteins in a particular transcriptome or a genome, it'll provide multiple files as an output. So it contains a statistic folder with all basic information regarding the predicted mitochondrial proteome, sequences, targeting signal analysis, so on, so on. It'll also give you every human mouse C. elegans Drosophila yeast ID, protein ID, for which it has and doesn't have a ortholog. So you can literally take that file, enter it in any of the functional analysis tools like consensus path and stringdb, and do functional analysis of your predicted mitochondrial proteome. It also provides an R-Shiny applet, which directly loads all this comparative analysis. It's kind of similar to the one which I showed you before, but doesn't have so many features in there. But it still lets you do this sort of comparative analysis of known mitochondrial, experimentally characterized animal mitochondrial proteomes and the predicted mitochondrial proteome of your animal species. Again, its R-Shiny applet was for the visual exploration. And so overall, this was just an introduction to what I did in my PhD. So that was kind of the first part of the talk, and then very briefly at the end, I also feel that I wanted to add stuff which my committee, my mentor, and my peers taught me in the US regarding doing research, or especially in PhD, because I can only talk about what I have done. So I definitely want to be an academic. I want to go into academia. So everything I do is from an academic point of view. But there are two main branches where, if you want to do bioinformatics in the US, of course, industry and academia. If you are going into industry, there are multiple paths you can take. You can go directly from your master's, you can do your PhD and then enter the industry. And then you can also do your postdoc and enter the industry, but it's less preferred. But interestingly, there are some really kind of very informative, not informative, sorry, very how do I say this? Very good industrial postdocs offered by companies like Monsanto and so on in machine learning and so on. So you can actually do an industrial postdoc and enter industry as well. In academia, PhD and postdocs, maybe multiple postdocs, but that's typically the path of entering academia. Very important thing which my co PI told me about doing a PhD, especially in the US, is that especially when you do your PhD, your goal is to learn how to do independent research, how to come up with a question, how to find resources to answer that question, and then through collaboration, answer that question yourself. So that the way my co-PI told me, this is not, your PhD is not for winning a Nobel Prize. It's for you to learn how to do independent research. And that kind of really changed how I looked at uh, PhD throughout, basically in my NS. And again, some very important points which I thought were important for applying for PhD in the US is something which I feel it's very important is first follow the research, not the university rankings. For instance, Iowa State University has some of the best statistics and biostatistic programs in the country, but you may or may not have heard of the university. Next is that most PhD positions are fully funded. Never accept a non-funded position, in my opinion. But it's not typically an issue in the US. Other countries, it kind of is, not in the US. And the whole idea is for you to find a lab where you want to work with, contact them. Don't hesitate about contacting anyone. Just send an email to labs where you want to work in, ask if there are PhD positions available. Is there funding? There are many, many ways you can get funding in the US. You can teach, you can be an RA, your PI, whoever you contact. If the professor is interested, that professor will find funding for you. That's what I can say. Another thing is uh, apply to a university where there are multiple labs you want to work in. You should always join a university which allows you to do rotations in multiple labs, which may help you select. It always helps, unless, of course, again, everything I say is which I found helpful. So yeah, and so that's just something to keep in mind. A very huge advantage of doing PhD in the US, that's something which I do want to talk about, is that if you're truly interested in doing a PhD in bioinformatics and you feel you lack the skill or the background, or you can learn these things in the initial stage of your PhD. So my first Hello World program was in the first week of my PhD. I learned Java, Perl, Python in the first 2 years, and R in the first 2 years of doing my PhD. The idea is not that I'm not kind of bragging about what I did. What I'm saying is that in US you have some time where you can actually pick up these skills. All your PhD committees want to see is that you are interested in research, you are interested in bioinformatics, and there are many ways for you to pick up these skills. So I wouldn't worry about if you lack any skills or background, you can still do a PhD in, say, structural bioinformatics. You may not have worked with structures before. You can pick this up in the initial stages of your PhD. So I definitely something which I wanted to talk about. And probably the most important thing is funding. Yeah. Uh, I taught for every single semester in my PhD. It's not something I would recommend. It's also something I would recommend, but it's important to keep in mind that there are multiple sources of funding, but the most common are either a research assistant or a teaching assistant. And there are pros and cons of both. Research assistants, you have more time dedicated to research. There's no teaching and grading. And con is you usually join a pre-existing project, a predefined project where you work within a particular framework. When you do a teaching assistant, the pro is that you have a lot of independence in formulating your own project. And it's very important to learn how to come up with a good scientific question. That's one of the most important things you can learn in a PhD. The con, well, teaching takes a lot of time. It takes a lot of time. It's really nice, but does take some time. But I would highly recommend if you're going to academia, make sure you teach at least for a couple of semesters. It really pads up the CV. So these were the other very important aspects I got an exposure to in the US. First was definitely teaching. So I usually took my students out to show them biodiversity. There was some nice beetle infestation on that plants in this summer, so I took them out to show them. But also, US lets you also increase your skills in science communication, which is very important, I feel, these days. It will give you, got I did a course in science communication and had many meet a scientist events. So here I'm in. So whatever I explain to you right now, I'm actually explaining this to these two, trying to explain some of this to these two kids here. So this was a sponge. This is a mitochondria. I had an activity to show how proteins work inside the cells. And so uh, with that, I think the very last thing I would want to end this was with postdoc research. And postdoc research is slightly not slightly, it's very different than PhD research. You typically do join a project. Here you are trying to basically, if you're going to academia, you are trying to learn how to work in a lab setting. You want to focus on learning how to get grants, get funding, manage, mentor students. That's what a postdoc is, as opposed to a PhD. Because right now you have learned how to do independent research in your PhD. In your postdoc, you're learning how to be a PI in the future. Takes some time. Again, apply for a fully funded postdoc, but you can also apply for funding. There are many different sort of places where they maintain lists of funding. And I think there is a way to send an email on this Bioclues thing. I'll ask Apan or Prash about this, but there are some really good resources about funding which I can't share here, but there are very nice detailed Excel sheets maintained by like Harvard and other universities, which I'll definitely share. And you can look at what sort of funding is possible. Problem with applying for funding is most of them requires citizenship in that country, so it's tricky, but you should always try. Currently my Microzone project is from funding, not so it's basically a grant, not given by the professor. Last thing I would say is join Twitter. Don't like, I know it's a very weird thing to hear, but Twitter is really very good. I found both of my postdoc opportunities from Twitter. It's really good. Trust me. Go to Twitter. Don't waste your time with like politics and time wasting stuff. Follow scientists, follow labs to make sure that Twitter suggests you those things. And trust me, Twitter is going to be your best friend while trying to find postdocs and even PhD positions. And the last thing is definitely something which also very important to keep in mind, consider countries other than US. Definitely consider countries other than US, Canada. I for my postdoc, I considered there were amazing opportunities in UK, Canada, Spain. Because of COVID, it gets complicated, but hopefully everything should dial down soon and you'll have access to many more opportunities in other countries. But I highly recommend that you look at where your research is as opposed to which country you want to go to. For instance, Canada has much better sponge research than US, which I found out much later, but not too late. So yeah, definitely look at other countries. There's some amazing research opportunities outside of US. And before just looking at the questions, I did want to give acknowledgements, non-family acknowledgements to my PI, Dr. Denis Lavrov, and my committee members, the Lavrov Lab, and these are my collaborators for my first postdoc, and my funding sources for my PhD and my first postdoc. And yeah, I thought I had it. Yeah, I guess that's the kind of entire presentation or talk sort of I had. So yeah, so I see one question. It says for PhD aspirants, are any specific sites to look? Yeah, so for PhD aspirants, I am currently mentoring an undergrad for looking for these PhD positions. Again, I would say first, Twitter is good. But next is, I would say you should look around for labs you're interested in. look out for research which interests you. Look at papers which are typically published by which professor publishes typically papers in that field. You can even contact the professor directly and ask them if there is a position available in their lab. But I would highly recommend that you search for this not on I don't think there are any specific sites which are very good to look for. LinkedIn has some PhD positions typically put from time to time, but it's not, by and large, it's not complete. So for PhD, I highly recommend. For PhD, you should definitely look for research you are interested in. So by that, you should also have a list of labs you want to contact. So yeah, I think that's unfortunately as specific as that would get. Yeah, but yeah, definitely. You should definitely Google is your best or best thing for looking for labs. Best way is to follow papers. Basically, look for which papers you are typically reading for in your field, which labs are working in that, and then contact the lab. Suppose. Uh, yeah. So if you are there and if you have any questions, I'm kind of wondering how to share. Let me share my email address, or I can email. I'll talk to someone to see how I can share my contact information. And you can always contact me if you have any questions, specific questions about like how to look for PhD positions or postdocs, because I literally went through that some time back, so it's kind of fresh in my mind. Yeah. So, yeah.
Thank you, Dr. Viraj, for such an informative talk.
Yeah. Thanks a lot. Thanks a lot for giving me the opportunity to talk. I like to talk about mitochondria a lot. So it's always nice.
Great.
Right. So, yeah. 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 Mm. Yeah. Oh, that is a very important point which you brought up. Is so the research just showed like all of that experimental research comes from one lab. So all procedures, everything, SOPs from one lab in Harvard, uh, Dr. Vamsi Mutha's lab. And it's kind of amazing that it's a question which troubled me also because it seems unbelievable that we don't have a standard procedure or even know how many proteins are in the mitochondria as such. And a lot of it also has to do with funding. Although although I have to say that in recent times it is improving. At least funding for mitochondrial research seems to be improving. But initially, when I joined, my PI always mentioned that it's kind of very tricky to get funding for basic research. And it's kind of that's what you mentioned later. It's more so a lot about emotional decisions by uh, the agencies, which is that the more funds go towards kind of more disease related, like something which has direct impact on diseases as opposed to something which could provide a framework for answering questions related to solving these diseases. So that's kind of the way I would put it. It is improving, guys. So, there are certain communities which are being set up as in the US. At least when I left, there's also but it's still not, unfortunately, it's actually shockingly missing in the US as well. So, and I'm in Canada, I haven't seen that much, to be honest. Canada is much more split up, which I feel. But yeah, unfortunately, that's the best I can answer that. It is definitely required right now. Uh, and Yes, to give you an idea, we like I the first postdoc which I was working on, we were looking at the same thing, as in uh, we are looking at DNA repair in mitochondria, which is kind of known-ish, sort of. It's not completely known how what all pathways are functioning, but it became difficult to write that grant uh, without kind of linking it to aging. So it kind of becomes tricky to. It's again a lot of science grant funding is always emotional. I feel. Sorry if I kind of went on a rant, but uh, I feel, yeah, that's what I feel about that. Yeah.
Thank you, sir. If anyone is not having any other question, then we can end the session.
Oh, sorry, there was one small question here. I just would want to
Okay.
talk that, but it's a brief question I saw. Is the same procedure for plant proteins? Actually, the answer is yes, because plant plants. There's a lot of research in plants. There's a lot of research in yeast, mitochondrial proteomes. The Arabidopsis mitochondrial proteome has been very well studied. Uh, I I can share my information. I think I should be able. Is it possible to share my information somehow through the list if anyone wants to ask specific questions? Because there are many that the procedure for plant mitochondrial proteins has been very well studied, and if you can contact me later, then I can actually send a paper on it because we were trying to actually replicate what's done in plant mitochondria in animals. So the plant, like, I think plants, there are multiple plants with experimentally characterized mitochondrial proteomes. Yeah, but not so much in animals. Yeah. Mhm. Sure. Oh yeah. Perfect. Yeah. Mhm.
Thank you, sir.
So, thank.
Yes, sir. Sir, actually, my PC's camera is not working.
Nice to meet. Yeah. It's really nice to meet all of you. And thanks a lot for giving me this opportunity. It's always nice to talk. Thank you. Thank you. Yeah.